El-Kebir from the University of Illinois. He will be talking about SPhyR, is that right?
SPhyR.
SPhyR. Okay.
Oh, wait. No, that's not a method. SPhyR.
SPhyR, okay.
All the single-cell methods are kind of like alike.
Which is tumor phylogeny estimation from single-cell sequencing data under loss and error. Thanks.
Okay, thank you for the introduction. So my name is Mohammed El-Kebir. I'm an assistant professor at UIUC, and I'm going to be telling you something about this work which got accepted at ECCB. So I think we've seen all of this. I can be quick about it. So the clonal evolution theory of cancer, so it starts from a healthy cell, you get a mutation, you get additional mutations, and in the end you get heterogeneity. And this process can be represented by a tree. And essentially what we want to do here, since this is the single-cell cancer genomics session, we use single-cell DNA sequencing to measure cells, individual cells, at the present time. And from these cells, we want to reconstruct the rest of the tree. So that's basically the setting. So in order to do this, you need an evolutionary model that tells you how somatic mutations arise in a tumor. So when I say somatic mutation, there's actually, they occur at different genomic scales. So most of the time, when people talk about somatic mutations in cancer, they talk about SNVs. So these are single base changes, single nucleotide changes. However, there's also copy number aberrations, where you lose entire chromosome arms. You also have whole genome duplications. And what's really important is that there is an interplay between these different types of events. So for instance, what could happen is that you could get an SNV, so a single base changes, so this G over here changes into a T. So you go from 0, which is the normal, to 1. And then you have a completely different event, like an indel, or a larger chromosomal loss, where you delete the mutation. So this is not a back mutation, this is a loss because of a copy number aberration. Now if you think about SNVs, so the number of SNVs in a tumor depends on the tumor type. But what we see overall is that most of the SNVs are actually just passenger mutations. They don't have any selective advantage for the tumor. So there is no selective pressure to actually keep the majority of the SNVs. So there's nothing that prevents this actually from happening. So this is something that's very common in tumor evolution. And if you want to have a proper evolutionary model, we need to take this into account. So current work, especially in bulk sequencing, but also in single cell, makes the infinite sites assumption. So in the infinite sites assumption, essentially what it boils down to is that there's no homoplasy. So that's a very fancy term. Essentially it means two different things. So first of all, it means if you look at an individual SNV, you can trace it back to a single cell where that SNV was introduced. So that means that there's no parallel evolution. And there's going to be a talk later this session, I think, even about this, which is kind of interesting. And the second assumption is that there is no loss of SNVs. So every descendant of the cell that acquired the SNV will have that mutation. So you go from 0 to 1 only once, and you never go from 1 to 0. You never revert back. So this is underlying site, which is going to be presented later by Katarina Jan, and also Onkenem from Florian Malkowet's group. So what I was just describing is that copy number loss is a very frequent event. So this is something that must be taken into account. And that's basically what I'm going to be talking about. So I'm going to split my talk into two parts. So first I'm going to assume that we have perfect measurements because I really want to understand what's going on computationally. And then I'll start dealing with real data, but we have errors in our measurements. So we've seen this before the break. So let me just briefly summarize this. So what I'm going to do is I'm going to assume that mutations are introduced only once. So you go from 0 to only once. But you can lose it k times. So this is Dolo phylogeny. And the Dolo restriction is about back mutations. So when I say back mutation, I actually refer not to reverting back at the nucleotide level, but actually a completely different event where you have copy number loss. So that's very important to take into account. So just formally, what this really means is, so our leaves correspond to the cells that we measure, and essentially a 0 corresponds to the absence of a mutation, the absence of an SNV, and a 1 corresponds to the presence. so we can, essentially what we want to do is we want to reconstruct the tree from these leaves. And a k-dollar tree can be defined very beautifully mathematically. So essentially, you can just capture with math what I was describing. So you go from 0 to 1 only once, and you go from 1 to 0 per character at most k times. That's essentially what's here in math. So we don't have to dig into that. And the problem that we have is we're only given the leaves, and we want to reconstruct the rest of the tree. So we're given a binary matrix. Rows are cells, columns are SNVs, and we're given a parameter k, where we have at most k losses per SNV, and we want to decide whether this matrix encodes leaves that form a phylogenetic tree under the k-dollar model. And if that's the case, we want to reconstruct this tree. So that's the computational problem that I'm going to be talking about. So I'm just going to give you the main theorem. So essentially what I did in this paper, I started thinking about this problem, like what's really going on. So if you think about the perspective from one mutation, essentially what happens is the following. So you start in a normal cell, you have state 0. And state 0 in your input actually corresponds to one of two different states. It corresponds to the absence of a mutation, and no ancestor of that cell had the mutation. So this is really state 0, the ground state. Or it could also correspond to a loss, where essentially you lost the SNV. So what you can do is you can actually think about is as an evolutionary tree at the level of a single SNV. So you start at state 0, then at some point you get the mutation, you get the SNV, and then you can lose it k times. So there's going to be k additional states, numbered from 2 to 2 plus k. So these are the lost states. And essentially what the game becomes is you're given this matrix with perfect measurements, and for every 0, you need to decide whether it's a real 0 or whether it's a loss. So it becomes an integer matrix completion problem, where from B we want to get this integer matrix A such that you encode a tree. Now the question becomes like, which matrices A actually encode trees, which is not a trivial question. So what I did in this paper, I basically have a complete combinatorial characterization of which matrices A, integer matrices A, actually correspond to trees. So if K is 0, then this matrix is disallowed. I think this matrix, for the experts here in the room, this is the 3-gamete condition. So if K is 0, you have perfect phylogeny, so no loss of mutations, and this is basically the matrix that's forbidden. But if you allow for losses, then there are 25 3 by 2 submatrices that are forbidden. And this number increases in the order of k to the 4th. So a simple brute force approach wouldn't work already. And this is actually a big open question. So I don't know what the hardness is of deciding whether an arbitrary binary matrix actually can be resolved. The zeros can be resolved such that you don't have any of these forbidden matrices. So that's contribution 1. So we have this complete combinatorial characterization of the solution space. So if you look at this number, so what I said is like that brute force doesn't work, but maybe we can encode everything as an integer linear program, which is a very prominent technique that's been used a lot in CompIO. So if you do a naive approach, where essentially you, as your variables, as your integer variables, you have matrix A, so the dimensions, the number of variables that you will need, is in the order of m, number of cells, times number of SNVs, times number of losses. And the number of constraints that you need to encode as forbidden submatrices is this number over here. So this doesn't scale in practice. So what we did is, I came up with this approach where you only introduce constraints and variables as you need them. So they're introduced on the fly. And this works well for practical problem instances. So we can solve practical problem instances in minutes, basically. And you can kind of like see that only a small fraction of constraints is introduced. So we're less than 0.001% is introduced. And same for variables. So this works very well in practice. Okay, so what I was doing was error-free data. So this is not something we can do in practice. So with single-cell sequencing, there are actually 2 types of errors. So there are, so some of the ones that you observe could be false positives. You could also have false negatives because of allelic dropout. And you could also have missing entries. So this is something that you must account for. And what I'm posing here is a problem that actually takes as input this noisy matrix D, where you have question mark entries, incorrect zeros and incorrect ones, but you don't know which ones are incorrect. And in addition, you have more information. So you know the false positive rate, and you know the false negative rate. So that's alpha and beta. And what I'm doing here is if you think about the evolutionary history of a tumor, so like what Florian Markowitz was saying, the units that really matter are clones. So if you have 100 cells from a sequencing experiment, you don't expect to see 100 different clones. You only expect to see a handful of clones. So that's actually what I'm going to do here. So I'm going to assume that there's only a handful of clones, only S clones. And in addition, if you think about your mutations, your SMVs, most of them will be passengers. There's only going to be a handful of drivers. So what this actually means is, if you think about the clonal expansion, so essentially what happened is, before the driver, you had a whole bunch of passenger mutations. So this corresponds to a long branch in the tree. So there's also going to be clustering in terms of the SMVs. So that's this parameter T. And this actually is beautiful because it allows you to really hone in on what really matters in the evolutionary history of a tumor. And that's what I'm doing here in this problem. And I'm using, so it's a maximum likelihood. So the objective function is maximum likelihood. I'm trying to estimate this smaller tree, this more compact representation, under the K-dollar model. I'm going to skip over this. So basically, so the summary is, it uses all the combinatorial characterization that I derived for the perfect case. So in general, when you have a hard computational problem, just drop the complexity of noise and then think about what's going on, and then you can incorporate that when you start dealing with noise. So that's essentially what I did here. The method is called not SPhyR, not SPhyR, it's called SPhyR, single cell phylogeny reconstruction. So how does it perform? Oh, that's too bad. Okay, so let's just look at these two plots over here. So what I did is I simulated trees with 50 cells and 50 SMVs. And the trees per SMV, you had at most one loss. And I used a false positive rate of 0.001. And I also used a false negative rate, beta star, of 0.2. Now, if you think about a Strawman method, where you don't change your input, so you basically output the same input, then what you will get in expectation is a false positive rate of alpha star and a false negative rate of beta star. So whatever method you run, it better be able to improve that. So it should do better than just like keeping the input as is. So what I'm comparing against here is SITE and SPhyR. So they use the perfect phylogeny model, the infinite sites assumption. And essentially what you can see is that there's a trade-off between a false positive rate and a false negative rate. So SPhyR, what essentially happens is it basically keeps the input unperturbed, because it has this evolutionary model where you can have homoplasy, where you can have many introductions of a mutation, parallel evolution, and many losses. So it's actually able to keep the input as is, basically. So that's essentially what happens here. And if you compare SITE to SPhyR, you can basically see that SPhyR is able to lower the false negative rate. And that's because of the dollar model. So it's able to resolve zeros as losses rather than something that needs to be fixed, needs to be a 1. So it can keep zeros as is. That's essentially what's going on here. Now, if you look at the trees, so this is just the matrices that were output. So just these are like the genotypes. Now, if you actually look at the tree, how well you cover the simulated tree, there are 3 different measures. So you could look at the fraction. So this was actually in the keynote by Sheng signing up. So there are 3 different measures. So if you look at a pair of mutations, a pair of SMVs, you could think about 3 different scenarios. So either one of them is going to be ancestral to the other, or they will be clustered together, or they occur in different branches. So there's 3 different measures you could use to measure similarity. Now, if the recall is 1 in all 3 measures, you recover the tree. So that's essentially what I'm showing you here. I'm showing you recall in terms of ancestral relationships, in terms of distinct branches, incomparable, and in terms of clustering. So what you can see is that SPhyR outperforms the other methods in terms of all 3 measures. So in summary, what this means is that SPhyR is able to reconstruct the simulated tree due to the dollar model, and also, importantly, due to the clustering that it uses internally. So this cluster is actually done not as a pre-processing step, it's done as part of the method. So it's a joint clustering and tree inference method. So in real data, so I looked at a colorectal patient described by the group of Nick Naving, and the resolution is really bad. You can still see the colors. So here I'm showing you the tree that was published in the paper, and this tree was inferred using SITE, which uses the perfect phylogeny model, the infinite sites assumption. And what I want you to look at are these blue cells over here. So these come from the metastasis. So there's a liver metastasis in this patient. And you can see that there are, and also here, there's a lot of blue cells that are part from the liver metastasis. And essentially, when you run SPhyR and you use this relaxation of a no loss, then you can actually resolve the metastatic clade better. So the blue cells, they appear here. And that's because of SPhyR's ability to recover the losses. So to summarize, I've shown you the combinatorial characterization of the K-Dolo model. And the beautiful thing is this model is able to strike a balance between being realistic and yet sufficiently constrained. So if you go for maximum likelihood or a finite sites model, you don't have enough constraints to deal with the noise in your data. So you need to constrain it a little bit. So that's what we did here. And I've shown you that solutions are integer matrix completions. And I've shown you results of SPhyR on simulations. And for future work, what I would like to do is I would like to incorporate alpha and beta into the problem statement. So I want to simultaneously find the false negative rate and the false positive rate. So right now what you could do is you could estimate these from germline SNPs. But it would be better if you could just do everything with fewer parameters. And in addition, I want to think about doing model selection for the number of losses and also the number of clones and the number of clusters. So in terms of, so essentially what is going on here, this is also a bigger problem with bulk sequencing. You don't know what ground truth is. And what's even worse is if you have an objective function like likelihood, there will be many different solutions that you can infer from the same data. And this must actually be represented in your solution space. And you actually have to think about how to deal with this. Like what are additional experiments that you can do to resolve this ambiguity? So I showed you one of the things that you could do. So if you have metastasis data, you basically want to use migration information to keep the metastasis cells together in the tree because you expect monoclonal seeding, for instance. So you need to incorporate additional information. And additional information could also be, for instance, what we saw this morning, RNA-seq. So I think that's where the field is headed. This is what we have to do to actually reconstruct the evolutionary history of the tree. So we cannot just rely on DNA-seq. We need to incorporate additional information. And for me, on a personal level, what I'm really intrigued by is just the hardness. I want to know what this problem actually is all about. So that's pretty much it. Thank you.
All right, any quick questions for Mohammed here? Yes.
I think it's great that the infinite sites assumption is being relaxed. And perhaps you're going too far. I mean, I wonder how often mutations are actually lost. I know in cancer. I know it happens some of the time, but it seems you are, in your model, this is even a likely event.
Yes.
And it might simplify the problem to basically realize that this is not a very likely event.
Yes, that's a very good point. So this actually alludes to my first point here. So you want to be realistic and yet sufficiently constrained. So maybe so what you could argue is for certain tumor types, you don't see a lot of copy number aberrations. And in that case, you could actually introduce additional constraints where you only allow a handful of losses and then play with that as a parameter. So that's something that can be done with this model. And I think this is actually something that needs to be tumor specific. And if you have copy number information from bulk samples, that's something that you should incorporate in here as well. Yeah.
Thank you.
Thanks.
All right, let's thank Mohammed again.